attempt over the weekend to run a two keys test, two keys on a significant difference test, because we hadn't done that yet on a two-way analysis of variance. For the two data sets that we've been primarily working with, one data set was the rat-fat data set, which is a replicated two-way design. The other data set is the supermarket data set, which is a two-way unreplicated design. So the way in which you do these things might or might not be different, because one is a replicated design and one is an unreplicated design. So hopefully you have the rat-fat data frame available to you. You have the supermarket data frame available to you. Now, if you do what I normally do, which is just call all of my data frames DAT at this point, this is going to cause problems because we're going to be flipping back and forth between these two data frames. So just be aware of that. When we move from one data frame to the other data frame, you're either going to want to rename your data frame something unique, or you're going to want to make sure that you load that data frame into R at the beginning of the next analysis. You can do it either way. It doesn't really matter to me. But just note that if you don't make sure that you're working on the right data frame, when you go to specify a model, it's going to give you errors because it's asking for supermarkets and you're in the rat-fat data and there are no supermarkets in the rat-fat data. So who successfully completed the two keys analysis for the rat-fat data? Yeah, what'd you get, Logan? Tell me your syntax. Is it two key test? Just all one word? Capitalized? No capitalization. Two key test what? Oh, gotcha. Assignment operator, two key. I was going to say that's not the way you do it. And then what'd you have in parentheses? Okay, and so what is model one for you? So whatever your data frame is called. In your case, it's that. All right, and that works for you. Okay. Did everybody else do something similar? Yeah. It seems pretty obvious. So let me get caught up with you. Make sure I'm in the right place. I have changed mine to rat just to keep myself squared away. So this is the analysis that I have. And so when you come down and you run two keys analysis, what you get is a bunch of combinations. So what the output gives you when you run a two keys on a significant difference test in R is the first thing that does you is it gives you the comparisons of the main effects. It basically says, is rancid overall different from fresh? And it is. Then it says, is sex different from each other? Is male different from female? And this is not. This should be exactly what your two-way Anova showed. So if we go up and look at the two-way analysis of variance table, it showed a significant effect of fat type and a non-significant effect of sex. And when we look at the F value and the P value, you'll see that the P value for this comparison is exactly the same as the P value for the result from the two keys test. Same thing for sex. There's the P value there, and it is 0.146036, and there it is there as well. So then we have this interaction term, and we want to know if there's an interaction, and there is no significant interaction. And so really when we look through this table of fat by sex interactions, we should really only see differences in rat type. So, in fat type rather. So when we look within females, the difference between rancid and fresh is significant. When we look within females and males, if you're comparing fresh and fresh to one another, they're not different. If you're comparing rancid and fresh, they are different. Fresh, rancid, different. Rancid, rancid, not different. Rancid, fresh, different. So in the case of the Tukey's analysis, all pairwise comparisons are all combinations of sex and fat type compared to all other combinations of sex and fat type. But the only ones that end up being significant are the ones where the fat types are different. And this is exactly what you would expect because there is no interaction term. So this is the correct way to test post hoc comparisons in a two-way analysis of variance. But once again, this is just the main Anova output. This is just the main Anova output. This right here. What you're really interested in is you're interested in all of these pairwise comparisons. That's the whole point of doing the Tukey's test. And once again, the result of the Tukey's test should conform to what your expectation is of the analysis that you did of the overall Anova. In this case, the only thing that is important is fat type. And so in those multiple pairwise comparisons, the only things that show up are situations where you're comparing fresh fat to rancid fat. And that's all there is to it. So that's how you would do that for that situation. And once again, this conforms to exactly what we would expect when we plot this. Did I already do that? I don't recall if I did that. So once again, those results should conform to kind of what we expect to see when we look at a figure of the data themselves. So when we go to the price data frame, who successfully did a Tukey's analysis for the price data? Supermarket data.
I might have that.
Okay, give it a shot. Okay, what was your analysis?
And then my analysis is AOV price and my tilde brand plus supermarket.
So in this case, your data frame is that, and I need two parentheses out beside that. Okay, so let's run that on these data. So for me, analysis 1 is the analysis that you specified, so I'm just going to go ahead and do that. It gives us a two-way Anova, but once again, the two-way Anova only has the two main effects and no interaction because all that interaction variability is in the residual term. And so we can do this. And so hopefully you got an output. So this is just, I could have just as easily put analysis 1 in here. But once again, when you look at your output, it's much bigger now because you have 10 supermarkets. But here, what you see is that the 10 supermarkets are compared to one another. Supermarket A to B, A to C, A to D, A to E, A to F, A to G, A to H, A to I, A to J. And then there's B to C, B to D, B to E, B F, B G, B H, B I, B J. CD, CE, CF, CG, CH, CI, CJ, DE, DF, DF, DG, DH, DI, DJ, etc., etc., etc. And you have the associated p-values with each of these comparisons. Now, it would be useful to actually look at a plot of this. So this is our not very pretty looking plot. And so one of the things that we should see when we look at this is if in our plot A looks different from B based on the means, the position of the means and their 95% confidence intervals, we should expect to see A and B being different here. A and B are different here. A and C don't actually look different here, but there's a reason for that, and I'll explain why. It shows it as significant here, but it doesn't look significant in this plot. A and D is not significant, and that's because A and D are virtually the same. A and E are significantly different. A to E looks like A's mean fits within the confidence interval of E's confidence interval, but E's mean does not fit within the 95% confidence interval of A's mean. And so when you look at these, the p-values associated with these comparisons don't match up necessarily to the differences that you see in the figure. And one of the reasons for this, actually the reason for this, is that these supermarket values are just values within that supermarket of the 4 brands. And it doesn't consider the fact that there are differences among brands when it calculates these confidence intervals. But you'll see that in your model, you have supermarket plus brand. So when you're testing means within a two-way ANOVA and you have both of the main effects specified, it's going to evaluate the variation of supermarkets given that it's already accounted for variation among brands, and it's going to account for variation among brands considering that there is variation among supermarkets. So do me a favor, go back up here to your Tukey's analysis and run the full model once, and then run the full model where you just look at brand. So just do price tilde brand. And what that will do is it will give you 2 analyses. One is the more complicated analysis up here, and it finishes off with brand. And then the other one just looks at brand. And so I want you to compare these 2 results to one another. In the first case, this is the lower half of the Tukey's analysis where you gave it price and brand. This is the analysis where you just give it price. It says, is there a variation among the different prices, among the different brands? Brand A versus B, A versus C, A versus D, B to C, B to D, and C to D. Those are all possible combinations. What do you notice about these 2 tables? They give you totally different results, don't they? In this table, the comparison between A and B is significant. The comparison between A and C is significant. The comparison between A and D is not. B and C is not, but B and D and C and D are. But in this one, in this case, none of them are significant. Why is that? Well, the reason that's showing up that way is that in this case, when you gave it the full model, it looks at variation among brands after it has accounted for variation due to supermarkets. In this case, because you just specified brand variation, it treats supermarkets as if supermarkets don't exist. Supermarkets are now just replicates of brand. And as a result, all of this wide variation among supermarkets gets rolled into residual variation due to brand. As a result, that residual variation within brand is really big, which then makes the denominator of your F statistic that you're testing really small. And so you get a different result when you just specify the single brand, because it then treats it as a one-way ANOVA, with brand being the main effect, and the data, the variation due to supermarket is just residual variation that's unaccounted for by anything. But it's not random variation, it's systematic variation that is due to supermarket variation. And so up here, where you gave it both supermarkets as a source of variation and brand as a source of variation, there are a lot more comparisons that are significant because it evaluates supermarket variation considering variation in brand, and it evaluates brand information having considered that there is variation due to supermarkets. Here, the variation due to supermarkets is simply residual variation. And as a result, it doesn't realize, the analysis doesn't realize that there's all this systematic variation due to variation among supermarkets in evaluating brand. This all gets thrown in as residual. And so you get a bunch of non-significant results. So what's my point with all of this? My point with all of this is that when you go to do things like post hoc comparisons, these comparisons after the fact, the model that you use is very important. When you have a two-way ANOVA, what you're basically telling R is that treatment 1, supermarket, might have systematic variation associated with it. Treatment 2, brand, might have systematic variation associated with it. Those two things may be independent of one another, or those two things may interact with one another, but you're telling it that there are these two main effects. When it evaluates the effect of one of those treatments, it considers the fact that there may be systematic variation due to the other treatment that you have in your data, and vice versa. And it actually removes that variation out of the analysis when it evaluates brand. So it removes variation due to supermarket when it evaluates brand. It removes variation due to brand when it evaluates supermarket. It doesn't just throw all that variation into the residual term. So in this way, it's also important sometimes what order you put your variables into the analysis in. Because in certain types of analyses, it accounts for the first term in your model first, and then evaluates the second term in your model, assuming that the first term has already been entered into there. In this case, it doesn't matter. We could go up here. Let me find my code again. We could go up here and run this data this way and get the analysis. But we could also go through here and just copy this and paste it down here. Make this analysis 2, changing the order. So brand comes first, and then we add supermarket. Why do I always hit the O instead of the P? And then this would be the summary of analysis 2. Oh, it is missing an A, isn't it? How did that happen? Oh, I didn't highlight it all when I did it. So what we see when we do these two things, here's analysis 1, here's analysis 2. Nothing has changed in terms of the mean squares and everything else. The f values are the same, the p values are the same. But we're about to, after we finish all of this, we're about to step into regression. And when we do regression, that's one of those cases where the order in which you enter variables into the model does matter. And so just always be cognizant of that. So whenever I analyze data, I usually have thought out in my head before I do an analysis which variables are probably accounting for the most variation in my data, and I usually put those in first in my analyses. So I structure my analyses in kind of what my a priori understanding is of what's likely to cause the most variation first. And of course, you hope that your treatments are accounting for the most variation and residual is accounting for the least amount of variation. But within, if you have multiple treatment effects, you might expect some things to cause more variation or account for more variation than other treatment effects. So just be aware of that as you move forward. Any questions about this? All right. That kind of concludes two-way analysis of variance. So what I want to talk to you about now is what comes next. So I'm in the process of trying to get your stuff graded and back to you. Hopefully I'll finish that in the next day or so. Um, but I want to talk to you about the Gecko analysis that you did. So pull that up on your computers if you can. Either the assignment or your script or whatever. So in the assignment that I gave you for the report, I basically told you to concentrate just on body temperatures, and then I told you to combine these treatments into I skipped something somewhere. So I asked you to combine the temperature, sorry, the season and the locality into two different into a new variable called treatment, right? So how did we do that? We did that by making a new data frame, or a new column in our data frame, which we'll call treatment. And we did this by doing what? What function can we use for this? Paste, right? And is it a concatenate? I don't do this very often. Okay. Oh, yeah. Okay. And then that $sign. population. Did that work? Oh yeah, so there's the treatment. So then I can tell it to do this with treatment. And then you come down, and after doing all of your checking and everything of assumptions, you basically hopefully ran an analysis that ended up looking something like this, where your treatment was amazingly significant and you got a bunch of things that were different. Um, you hopefully got a figure that looked something like this, or maybe nothing like that. This is so this is a different type of plot. I should probably introduce this plot to you. So what I basically did was I've essentially I've thrown a lot into this one figure. And what I've done, so this is the figure that I would have generated potentially. But this plot tells us a lot of things. One of the things that this plot tells us is it gives us the means and the 95% confidence intervals for each of these different combinations of population and season. And I got that by running a confidence interval, running summary SE and making a little data frame out of confidence interval. But then the other thing that I did was I made a dot plot with the original data. So one of the cool things about ggplot is you can combine different data frames into the same plot, as long as, for example, you have the same x-axis variable. And so what this does really nicely, I think, is it shows you that not only are these two things different from these two things, and these two things are different from one another, it also shows you each individual observation jittered to either side of the main category, such that you can see some idea of what the distribution looks like. And you see that in this case, this particular treatment is skewed. A lot of cooler temperatures, and then some temperatures that are somewhat warmer than the average. And so when we saw that lack of normality, I told you that it was due to skew, and it's due to skew primarily due to this treatment and this treatment, somewhat due to this one, lesser so to that one. So there's this positive skew where you have some high values that stick out from lower values. And we accomplish this plot by simply adding a dot plot. And in this case, the dot plot, it isn't getting data from the summary SE output, it's getting it from the original data frame. And that's nice. That's a useful sort of thing to do. You'll notice that I plot the error bars and the points on top of all of that so that you can actually see the error bars and the data points. If I put the data points and the error bars further up in the ggplot, it would have put the dot plot on top of those and it would have been impossible to see them. So remember that R does things in order. It's just like any other program. It does one thing, then it does the next thing, then it does the thing after that. So you did this because the researcher told you that he wanted you to look at this combination of seasonality and population, and he was a simple-minded buffoon because he wasn't aware of what? Is the way you analyze the data in that first report, is that the way you should have analyzed the data? Why or why not? Critical thinking caps on. The researcher gave you the data in this form. You had to add this thing, which is a combination of season and population, whereas the original data came to you as a separate column for season and a separate column for population. Is the way you analyzed these data in the last report, is that the most appropriate way of analyzing these data?
Probably not.
Why not?
I just want to look at each individual variable rather than this.
Why not just look at each individual treatment effect? Why not look at season as its own thing and look at population as its own thing? And then what do you have the ability to do? You have the ability to look at both of them simultaneously, and you have the ability to look at the interaction. What came out of the one-way Anova looking at treatment is significant mainly because summer and winter are different from one another. That's one thing that comes out of it. But populations are also different from one another. But what's most important is that in this data set, there's an interaction between population and season, such that both populations in summer are not different from one another. But the populations in winter are different from one another. In Galveston, winter temperatures are much warmer than they are in Oklahoma City, because Oklahoma City is further north. And so what you should expect out of a two-way analysis of variance is you should expect a significant effect of population. You should expect a significant effect of season, but you should also expect a significant interaction effect. Because if we were to draw lines from summer Galveston to winter Galveston, and summer Oklahoma City to winter Oklahoma City, you see that the slopes of these two lines is different. Those two lines are diverging from one another as you move from summer temperatures to winter temperatures, and that divergence is a sign of a two-way interaction. So you analyzed this data set because a researcher told you a particular way that he was thinking about the data set. Because he didn't know about two-way ANOVA, he probably only learned about one-way ANOVA because he had an introductory statistics class that didn't get any further than that. You now know about two-way ANOVA. Had the researcher come to you and asked you to analyze the data in this way, it would have been incumbent upon you to say to him what? Let's do a two-way ANOVA. So much more interesting, so much more nuanced way of thinking about your data, because you really have these two sources of variation. You have a source of variation due to seasonality. You have a source of variation due to population. Let's see if those two things act independently of one another, or if those things actually interact with one another. And then he would have been like, no, this is the way that I think about my data. And then you would have had to convince him that your way is the better way. There was a time when I was younger when I thought there's one most appropriate way that any particular data set can be analyzed. The way you analyzed it in the first report is the way I asked you to analyze it because we had only been exposed at that point to one-way ANOVAs. Now you've been exposed to two-way ANOVAs, and even though there's nothing necessarily wrong with the way we analyzed it that first time, it's not perhaps the most appropriate or most useful way of analyzing the data. Now we know more about statistics. We have more tools at our disposal. And while that might have been a fine thing to do when all we knew about was one-way ANOVAs, it's not the most appropriate way. It's not that it's inappropriate. It's just that you lose some information. You lose the information about interaction. This happens a lot. This happens a lot in molecular biology. I occasionally get asked by molecular biologists to help them analyze data. And so they will have a group of cells that they're growing under some condition. So they have a control. where they're just giving them a particular temperature, they're giving them all the nutrients that they need to survive, and they're just growing. And a lot of times in this department, at William Jewell College, these are cancer cells because there's a group of people here interested in cancer biology. And so they're oftentimes testing different forms of treatment. And so they might have treatment 1, and they'll give, 0.5 millimolar concentration, a 1.5 millimolar concentration, and a 2.5 millimolar concentration of this particular substance. But oftentimes we know from cancer that this would be drug 1 at 3 different concentrations. So this would be like a single treatment at 3 different concentrations. And then they might do treatment 2. Once again, maybe this is a different kind of drug that gets administered at a very different concentration. So they might do this at 2 millimolar, 4 millimolar, and 8 millimolar. And then they bring this to me. And so the control is basically none of this, and none of this. So they bring this to me, and I'm like, oh no. And what's wrong with this as a two-way analysis of variance? If we were to take this as a two-way analysis of variance, we might create a graphic to describe this in this way. Drug 1, we have 0 of it, we have 0.5 of it, we have 1.5 of it, we have 2.5 of it. Drug 2, we have 0, 2, 4, and 8. And then we have observations of this. This is the control. This is the first treatment combination, 0.5 and 2. So we have a combination here. We have 1.5 and 4, 1.5 and 4, and we have 2.5 and 8. 2.5 and 8. Can we analyze this with a two-way analysis of variance? Okay, you say no, but you don't know why. Do we have all possible combinations of these two drugs? We don't. We have nothing. We have more of one drug and more of another drug, more of the two drugs, and even more of the two drugs. So while we might be able to assess the effect of drug 1 and the effect of drug 2, what can we not assess? We can't assess whether they interact with one another because we don't know what happens when we give these cells a lot of one drug and nothing of another drug. We don't know what happens when we give them a lot of one drug and a little bit of the other drug. And yet, the thing that they're interested in, the thing that they're coming to me for help with is they actually aren't necessarily interested in what's the raw effect of these two drugs on cancer cells. They're actually interested in whether or not these two drugs interact with one another. In many cases, the thing that they're most interested in isn't the main effects, because chances are we already know what the main effects are because they've already been tested by themselves with these cells. They're really interested in the interaction term. And yet, they've designed an experiment that makes it impossible to get at that interaction. Because in order to get to the interaction term, you have to know what happens when you have a lot of one and very little of the other, and a lot of the other and very little of the one. That's the power of a two-way analysis of variance. And if you design an experiment that prevents you from looking at that interaction when that's the actual interaction that you're interested in. You just wasted a bunch of time. And then you have this conversation with them, and it's like, wow, that would have been a lot of work to do all of that. I'm like, yeah, that is a lot of work, which is, of course, why you didn't do it that way. But what you could have done, had you consulted a statistician beforehand, is you could have said, well, I only have a limited amount of time, a limited budget, a limited amount of energy and sanity to put towards this thing. So what could I do? Well, maybe rather than having 3 levels of everything, you just have a low and a high, and a low and a high. If you get rid of the intermediate things, you take this whole thing and you reduce it down to 0, 2, and 8, 1.5, 2.5, 0, dot, dot. You've now reduced the possibilities. You were already going to do these 3 things in the first place, as well as 2 more, right? Because it was 1.5, 2.5, oh, and then 1 more to get the 8 and the 2.5 over here, because you had a 1.5 and a 4 in the middle. So you were already going to do 4 of these things, right? 1, 2, 3, 4. Now I'm just saying you have to add more complexity. So you're more than doubling your effort in the whole thing. But what you're gaining in doubling effort is you went from 4 treatments to 1, 2, 3, 4, 5, 6, 7, 8, 9. So you went from 4 treatments to 9 treatments, 2 plus doubling of effort. But what you get out of that is you actually get to be able to do the thing that you actually wanted to do, which is to test whether or not these 2 drugs interact with one another. And then they get to the end of the analysis, and you tell them that you can't tell anything about the interaction because of the way they set up their experiment, and they're disappointed in it. But they could have thought through this better at the beginning by thinking about what the analysis would be like in the end. The whole point of doing a 2-way Anova is oftentimes because you are interested in the interaction, just as much, if not more, than the main effects. Otherwise, you would have just been testing the main effects and made your life much easier. So doing things in ways and analyzing the data in ways that allow you to get at that interaction effect are important. And so you had a 2-way analysis of variance kind of set up that the researcher asked you to convert into a 1-way Anova, which seems counterintuitive. But this guy is not that bright, clearly. Not that experienced with statistics, and so you just went along with what he asked you to do. But now you know that there are better ways of doing this, and actually that's going to be your second report that you write for me. You're going to revisit these data, and now rather than looking at them as 4 different treatments, you're going to look at them as these different treatment combinations, where you can treat season independently of population, and you can calculate what the interaction term is like. The reason that I'm doing this is because I am the researcher. I am the one who told you to do the 1-way Anova on what is essentially 2-way Anova data. I want you to see in the process of looking at this data set twice what the advantages are of running data using a 2-way analysis of variance rather than a 1-way analysis of variance. And the best way to do that is to take a data set that is possibly analyzed both ways and see what the differences are between those 2 things. The thing that we're about to start after we're done with 2-way Anova, which this concludes the end of 2-way Anova, is we're going to talk about regression. Regression is a situation where you have one variable that influences another variable. And so Variable x influences variable y. Both of these variables are continuous variables. And as x goes up, y goes up, or as x goes down, or as x goes up, y goes down. First of those would be a positive correlation. The next of those would be a negative correlation. Or there's a third possibility, which is the null hypothesis, which is as x changes, y just is random, slope of 0. And so in this case, the hypothesis that we're testing is a hypothesis about the slope of the data. Well, you guys have been working on body temperature. And in that data file, I asked you to pay attention to body temperature and ignore all of these body temperatures. Well, in that data file also, there are wall temperatures. So this is a species of lizard that lives on walls of buildings. One of the reasons it's easy to study is all you have to do is walk around a city that has this species in it, and they're crawling around on the walls at night. Normally, lizards will thermoregulate. They'll be out there in the sunlight. When they're cold, they'll run into the sunlight. They'll bask on a rock or something. They'll raise their body temperature up. When they start to get too hot, they'll run into the shade. They'll cool down a little bit. When they get too cool, they'll run back out into the sun, and they'll shuttle back and forth. And they'll regulate their body temperature such that their body temperature stays pretty constant over time. But this particular species, it's a gecko species, which means that it's nocturnal. It occurs at night. It's active at night. So at night, running around on buildings in some of the skeeviest sections of cities that you can possibly imagine, these lizards are running around on walls. And what I do is I walk up to the lizard and I measure its body temperature, and then I measure the temperature of a spot on the wall right next to it. Now, if this was a diurnal lizard, the temperature that it is at might or might not match the temperature of the habitat right next to it, because it might be in a sunny spot, but it just moved there from a shady spot and is in the process of warming its body up. Or it might be in a shady spot because it just ran out of the sunlight to cool down in the shade, such that its body temperature is way higher than the shady spot that it happens to be in. And so in those cases, the body temperature of the lizard will be not correlated at all with the body temperature of the habitat. So if we make this habitat temperature and we make this body temperature, we might expect the habitat temperature to not be related to body temperature at all in actively thermoregulating organisms, because the organism is trying to maintain its body temperature at a particular level. But if it's a nocturnal organism and the opportunities for thermoregulation are reduced, we might actually expect what to happen? If the animal can't thermoregulate, its body temperature is just going to be what? It's going to be probably lower in general, but also what is it going to conform to? Yeah. And so what you might see in a nocturnal, we call these in biology thermoconformers, we might see a positive correlation between body temperature and whatever the habitat temperature is. If you are a thermoregulator, though, you might expect those two things to not be correlated, because the animal is actively doing things behaviorally to set its temperature at a particular level, regardless of what habitat it happens to be in at that particular moment when you measured its body temperature. So, same data set, an additional variable added in, but now we can look at temperature with regression analysis in a way that's completely inappropriate with an analysis of variance type of structure. So you're going to revisit this data set over and over and over again over the course of the semester. Not because I think you care anything about thermoregulation in lizards. Not because I think you care about ecology of lizards. Not because these things behaving differently in different environments that are separated by latitude has something to do with its ability to continue to expand northward, because it's an introduced species that started out in South Texas and South Florida and has moved as far north as Pittsburgh, Pennsylvania. Very cold environment for these individuals. I don't expect you to care anything about all of that. But I want you to revisit these data because it shows you that for any given data set, there are potentially a variety of different lenses to look at the data through. And each of those lenses corresponds to a different kind of analysis. So we're going to apply two-way Anova to the same data that you've already looked at. That should make writing the second report easier than writing the first report was, because you're already familiar with the data set. We are going to look at the temperature data using a regression analysis because partly you've already seen part of the data. You're already kind of familiar with the data set, but then it also gives you a different lens through which to look at these data. And once again, because you're already familiar with the data, writing the regression report should be somewhat easier than writing the first report was, simply because you're kind of familiar with the data. And all you have to cope with is the learning of a new analysis technique, rather than coping with both a new analysis technique and a wholly new data set. So that's kind of going to be the plan for the rest of the semester. We're going to think about this data set in different ways using different methods. So that's what's coming up. So also, the other thing that I'm going to do is as I finish grading your past assignments, I'm going to make 2 assignments today. I'll put them on the Moodle site later on today. First one is a short problem set looking at two-way Anova. Now that you know how to do things in two-way Anova, we're going to work out a two-way Anova by hand, just like we've done in class. And then I'm also going to assign the next report that you'll have due, and it'll be due in a couple of weeks so that you have time to work on it. My hope is to have your first report back to you in a couple of days so that you can look at feedback from that and use that in crafting your next report. But that's kind of where we're going. Questions about that? All right, rather than start something new today, I'm just going to let you guys go early because we could get into regression, but we wouldn't get very far into it. So on Friday, look in the daily schedule. I'm not sure how far off schedule we are, but we are somewhat off schedule. Find the section that deals with regression. Go and read your textbook on that particular section of material. We're going to start, like we always do on Friday, with a simple example that is easily worked out by hand. And then we'll move on to more complicated examples, leading us up to dealing with actual kind of real world data. All right, questions? We didn't really do any live coding, so don't worry about giving any feedback. And I will see you guys on Friday, because what is Wednesday? It is the final, I think it's the final spring break day, isn't it? Yeah, it's the final spring break day. So I will see you on Friday. Try and do something fun on Wednesday, preferably without interacting with humans. That restriction kind of puts a damper on it, doesn't it?